Okay, good morning. Fine, is that board visible? Good morning. Is that board visible? Okay, good morning. Good morning, everyone. So, yeah, great. So, today what we're going to learn is cell division and cell cycle, right? Cell cycle and cell division. Compared to the, what do you say? Comparatively to the first lesson, that is cell structure and cell function, this lesson signifies more, but the basis would be that lesson. So once go through the cell organelles also. Okay, if you have doubts you can ask me. That's other thing. But these cell division events would keep coming in your 12th standard portions also. So especially when we talk about the cell cycle, the different phases that are present in the cell cycle are utmost important. Okay, so here we need to more focus on this meiosis 1 prophase 1. So I'll try to give some diagrams of it, where during last year class also I gave with different colors, like how this homologous chromosomes would actually pair up, crossover, right? And at the same time they form the chiasmata, and then finally they recombine, right? Recombine and then separate, right? So these phases are important in this. So before we actually go into the meiosis, first we need to know about why this cell division. These are all lower standard questions, like why the cell division takes place, and what is the roles of what is the uses of cell division, where cell division occurs. For example, what are the types of cell division? We have mitosis and meiosis. So one of the cell division occurs in the one occurs in somatic cells and one occurs in reproductive cells. Okay, so what is the difference between good morning. So what is the difference between mitosis, meiosis? These are all things we already studied in lower standards, and you may not expect any sort of good questions from that. But what you can expect is, from, you would have remembered the cell cycle, right? So cell cycle comprises of following following aspects, right? What are the components of this cell cycle? What are it contains? So phases it contains, for that matter. If you divide this cell cycle, right? So this cell cycle, right, it can be of two phases. It will be two phases, right? So one is no we have two phases in this. One is interphase, right? And one more is M phase. M phase. So if you see here, for my CBSE students, I already gave some mnemonics to remember these M phase. What are the events that does it takes place? But whoever metric students are there and new students are there, there is a memory technique. Okay, there is a mnemonic here to remember. So within the M phase, you will be seeing P. I will be taking one more. So within the M phase, you will be having P, MAT, C. Okay, is it visible, guys? P, MAT, C. That. So what I'm saying is, whoever new to this class, this would be new. So P, MAT, C is nothing but prophase, metaphase, anaphase, telophase followed by cytokinesis. Clear? So cytokinesis is a end of the cell division where the cell would be divided, where cytoplasm would be getting divided, right? So P-maxi is a mnemonic for this M phase. So that is mitotic phase. Okay, and interface again will be having interface again is divided into G1S and G2 phases. So what is this interface? If you see this cell cycle, right, most of the cell would be actually spending more time. If you take the eukaryotic cell, it is almost 24-48 hours the cell requires to divide. So most of the significant time would be spent in this interface actually. So interface again we have right phases like G0 is something, I'll explain what is it. G1S, G2 phases, right? So these are the interface, and you have got M phase. So M phase occurs very quickly, right? But this phase only takes a lot of time. Why? Because each phase is significant roles are there within this each phases, right? So let's see, this cell cycle is the one of the thing that actually, we take like this cell cycle. So we have certain events that occurs in the cell cycle. So when the cell cycle is not proper, right? When cell cycle regulators, for example, we have certain proteins, okay, certain proteins, cell cycle cycling dependent and cyclic proteins are there, which actually controls the cell cycle. Okay, so my question is why cell cycle has to be controlled? Why can't we just leave it like that? Why we need to control the cell cycle? Why cell has to have a control on the cell cycle? If there is no control, what happens? Only today I will not ask more questions. So only one question I'm asking. Cancer, okay. What exactly happening? Cell cycle regulation. If the cell cycle regulation is not there, what happens is they try to divide. They proliferate. They keep on dividing. Actively, they keep on dividing. There won't be any checkpoint, right? There won't be any stop. But if you see our cells, right, our tissue arrangement, cells arrangement, if you see, there will be certain number of cells should be there. The number and the location also very much important. But more than required means they are tumorous cells. So tumor might be cancerous or not. So there are non-cancerous and cancerous tumors. So tumor is something which is unwanted mass of cells, tissue, right? So how come this tumor is forming? Because the failure in the cell cycle regulation, right? So that's what the cell cycle regulation and their events, especially the G1S, G2 phases are extremely important. You should be carefully focusing on them, right? So if you see that each phase, right, what exactly happens in G1 and G2 and S phase. So if you see this G1 and G1 phase, what happens is G1 would be most metabolically will be active, and it will be prepared itself to enter into the S phase, right? So this G1 phase cell, so I'm rubbing this. So we are talking about interface. Once we complete interface, we'll come to P-maxi. Okay. So when we talk about what do you say, G1 phase, right? The cell within the G1 phase would start growing metabolically. It will be active. At the same time, it will be increasing in its size also, right? Is that clear? So in this G1 phase accomplishes the most of their growth. So growth would be taking place in this G1 phase. So first we are talking about G1 phase, right? More growth would be there in this G1 phase. So it will be bigger in the size and makes more proteins and organelles needed for the normal functions of the DNA synthesis. So here what happens is different proteins will be made, right? at the same time, this entire cell would be keep on growing and it will be ready for the next phase, that is S phase. Okay. For example, proteins and RNA synthesis, right? These are all RNAs, everything will be synthesized during this G1 phase. So these cells are fully functional, right? Is that clear? In addition to being, being on a dividing platform, right? So because this is going to undergo division, right? Right? Though it is undergoing division. For example, look at this room. Okay, this room, if I wanted to partially divide it to two, okay, even so, what I will do is I'll try to bring all the equipment to construct a wall in this room or house. So even before constructing or during the construction, my living room would be functioning normally because I will be moving, I will be taking classes, board is there, everything is there. But even the division or construction is also going. So this G1 phase, the cell would be normally functioning. Though there are lot of changes, growth is there, size increases, proteins are formed, RNAs are formed just to prepare itself for the next cell. That is S phase, right? So what exactly happens in the S phase? You guys understood what I'm speaking of? Are you able to follow? Are you able to follow? So is the G1 phase clear? Yeah. So what exactly occurs in S phase? What do you mean by S phase? For that matter, S phase. So we are talking now about S phase. S phase or synthesis phase. What exactly happens during what happens exactly during a synthesis phase? DNA replication. So here the DNA would be just like thread-like structure. Yeah, Matyalana, Arshit Priya, Sri Durga, uh, yeah, correct. So here synthesis phase of nothing but the DNA would get replicated. So it doubles its content so that it can enter into the next phases, so that the distribution of this homologous chromosomes later part, that is during M phase, would occur. Okay, so for that matter it needs to double its DNA or the genetic material, so it undergoes replication. So replication is one of the important aspect of the DNA and RNA genetic material. And once it replicates, what happens is it enters into the next phase, that is G2 phase, right? And this within this S phase, this DNA replication occurs, and we know in molecular basis of inheritance we already studied during S phase. I mean during replication, what are the events does occur, right? So the replication fork would be formed. So here you don't need to focus in details of the replication, but you need to remember what are you have to connect to that, right? So during this synthesis phase, replication takes place, and what are the polymers you should be remembering? DNA dependent DNA polymerase, because based upon the DNA strand only, it is synthesizing new strand. So that's what's a replication, and direction of the replication also you should be remembering. Okay, so these are the S phase events where it undergoes synthesis of the synthesis of DNA. Okay, and what happens in the G2 phase? If you see in the G2 phase, G2 phase can be considered as second growth phase. Okay, first growth phase is G1 phase. Okay, so first growth phase is G1, and the next growth phase is G2 phase. Once the G1 is ready for made it prepared everything for entering into S phase. Now during S phase, along using the proteins already produced and RNAs, it started synthesizing its own DNA, right? And then it enters into the next phase, that is second growth phase, right? Which followed by the S phase, right? So here what we need to understand is it will undergo quality control checkpoints. So here it will be, it will be taking care of the DNA, integrity of the DNA, right? Whether the DNA is proper, whether there are any errors are there. So you might have remember the DNA polymerases, right? There are some exonucleases we have at the same time, exonuclease activity. There is proofreading mechanism. So here quality control would be occurring in the G2 phase. So during this second growth phase, this quality control or this quality control checkpoint, quality control of DNA would be checked. So then after this, the cell enters the secondary growth phase where the nuclear envelope also would be trying to forms. In this phase 2, here one more thing that is demarcated is formation of centrosomes. So in last class you would have, yesterday you would have revised the cell organelles. So there are centrosomes and you have centrioles formation, and the centrioles arrangement. They are perpendicular, perpendicularly arranged, right? So one would be like this, one would be like this, perpendicularly arranged, right? These are the two centrioles would be formed during this G2 phase. And now why it is formed here, the centrosomes and centrioles in the animal cell especially, because it is preparing itself for the next phase, that is M phase. So once the G1 as G2 phase is completed, the cell enters into M phase. Okay, so here we need to understand few checkpoints also. These are the things that happens in G2 phase. Even the microtubules formation, it means the microtubules that will be used to mobilize the chromosomes during M phase. So it is followed by M phase, right? So during M phase, division occurs, P mat C. So the chromosome has to move further apart. So that needs certain microtubules, centrioles, etc. So those are all things would be prepared in the G2 phase. Once it is prepared, it is now ready for entering. Now the cell is ready to enter into the M phase. Here we have certain checkpoints actually, right? For example, I told cell cycle checkpoints. Right, these are the checkpoints. These are very potential questions in NEET book. It was very they were very clearly mentioned actually. So here there are checkpoints that you need to focus. What are those checkpoints? So cell cycle checkpoints. Okay, what are the cell cycle checkpoints? Checkpoints in the cells. I told the cell which is, there is one more phase that is called G0 phase or quiescent phase, right? Where the cell will be freezed, in the sense like it will not enter into the any division. For example, if we consider neurons once formed, they will not again replaced by new neurons. It means formation of it would be static. The neuron would be static where it fails to enter into the next divisions, next phases, so that the neuron cannot really replaced, or once it is damaged, it's damaged. So why? Because of the cell cycle checkpoints. So here we have certain checkpoints like exit M and enter S checkpoints. What is it? We have exit M and enter S. Okay, enter S checkpoints. So even before the cell enters into the G1 phase of the interphase, it goes through the exit M checkpoint. So here you need to understand what are these checkpoints and why we need this checkpoint, why the cell needs checkpoints. You need to understand. So you have to always have a diagram in your running notes, the practice notes, so that you will not be confused. So put this G1 and S and G2 phase and put like M phase. Okay, M phase and G0 phase. So when you have this thing now, you can you can go back and relate and you can really understand. So even before cell enters into the interface. So what is the interface? This is the interface, no? G1s G2 phase, right? Even before it cell enters into M phase, it this interface, it goes to the M phase exit checkpoint. So here the cell is checked to ensure that it has completed the mitosis phase. So it means in between the mitosis, the cell would not be coming out. It means it will not cease its activities. It will not stop its dividing until or unless the cell is divided. Once it exits the mitosis only, then it will think of the remaining thing, whether it has to enter into G0 phase or G1 phase. Then it will be decided. So once the cell enters into the mitosis M phase, there won't be any stop in it. So significantly it will keep on going. So exit M phase means this one of the checkpoint is that before even entering the interface, the cell ensures that it completed its mitosis, right? So once the mitosis is formed, mitosis is happened, right? So we have got two cells. So now this two cells fate is now decided whether it has to enter into interface or G0 phase. Did you understand? Are you able to follow, guys? Are you able to follow? Exit M phase is clear, right? So exit M phase and this S center phase, I'm using new words. That's it. But during NEET class last year, I explained about these phases also, right? I mean, what are the checkpoints? But the words that I'm using right now would be new to you. Okay. So this is how this even before it is entering into the interface, it would be checked. Okay. And then next one is before the cell comes to S phase, it goes through the G1S checkpoint, also called as enter S phase. So now we are talking about enter S phase. So even before cell commits to enter into S phase, right? Whether the cell has to enter into S phase, okay, this way. So whether the cell has to enter into S phase or not will be decided at this point. So enter S phase. So that is what. So G1S phase we will say, that is the one of the checkpoint, also called as enter S phase, where the nutritional status of the cell would be thoroughly checked and even integrity of the DNA would be checked, right? So these are the important aspects that do occur during this enter S phase, and then only if everything is fine, then it will be entering into the S phase. But once the S phase starts, until it finishes the S phase, this will be keep going. Okay. Clear? So this is very important step, G1 S phase, because the cell once enter the S phase, it keeps on going actually. So that is one of the points. So this is one of the important point. So now we have to understand in the mitosis, what are the different cell cycle regulators we have. So these cell cycle regulators are of two types actually. Two. So enter point, exit points, those were already given in your NEET book. Once go through that. So here we have two groups of regulatory proteins are there. Okay. One group of the regulatory protein is cyclins, right? So you would have observed during writing NEET exams, I would have asked these questions also. We have got cyclins and cyclin dependent kinases, that is CDKs. Okay, so you need to know their functions also. So cyclins and CDKs, the activity of this CDKs fluctuates along with the cyclins. So these cyclins are the proteins that regulate the timing of the cell cycle. So how much the cell has to present in that particular phase would be decided by this cyclins, right? Even their levels also will be fluctuated across the cell cycle. right? So likewise, we have this different cell cycle regulators. If you see this, uh, for example, this CDKs actually again checks that whether this Pmax is occurring properly or not, right? So cyclin and CDK complexes would actually halts the progression into telophase. It means it will may not enter if there is any, if they wanted to arrest the cell, they will be arresting at that particular point, right? It will not allow to, separate, and it not allow the cells to enter into the next phases. At the telophase, they will regulate the cell cycle at different levels. Is that clear? Here, so here, which is a important checkpoint is G1 phase, where the G1 S phase entering into the S phase. So that is an important checkpoint that you can have, right? So like that, we have CDK proteins which actually regulates the our M phase also. So how come this, this, what do you say, in which phase this CDK proteins actually regulates? You need to remember that, once you go through that Arihant material, you will be understanding, especially the NEET material. We have talked about different CDK proteins, different cell cycle regulators, and their checkpoints that you need to go through. Is that clear? So we can move forward if you understand this, right? So you need to focus on these aspects. Cell cycle, it is divided into interphase and M phase. And M phase, you have a P-maxi, prophase, metaphase, anaphase, telophase, and cytogenesis. And then we have cell cycle regulation by cyclins and CDK proteins. Okay. At the same time, we have two types of checkpoints also. I have explained that also. You please try to focus on them. So when we talk about different types of so when we talk about different types of divisions, we have mitosis and meiosis. So far, what we talked about mitosis and meiosis is actually, if you would have remember again, it is divided into M1 phase and M2 phase. That is meiosis 1 and meiosis 2, right? So here the bonus of meiosis is that if you understand mitosis, meiosis 2 is the same. Okay, there is no change in any genetic components of the cell during meiosis 2. So you can put these words together. So I will be giving a few words that you can, it will make more sense actually. Okay. So for example, okay, right. So I'll draw a small flow chart. See if it is really understandable or not. So what happens is meiosis is a special type of division, right? It is it is observed by Farmer and Moore, right? Farmer and Moore, and it is a type of indirect division which occurs in diploid sex cells, right? So once a cell is undergoing meiosis, right? So what happens is it eventually enters into M phase and M2 phase, results in formation of 4 haploid cells. This is diploid, right? 2 and 5. So numericals, number questions may come from this type of a concept. We have gametogenesis, we have especially the oogenesis and spermatogenesis. We have already studied this, right? So that's what I always keep remembering you guys, that you need to go back and learn the 11th standard portions also. Okay. But whoever took this CBSE marks seriously and they left NEET in between, anyhow academics exams are over, so there is no headache. But this is how you need to prioritize. Okay. Anyways, if you see here, meiosis is a special type of division, and reduction division. Why we call it as reduction division is because if there are 46 chromosomes, it is becoming 23 chromosomes. There is a reduction in the number of the chromosomes, right? So during meiosis 2. So how can we depict it, and how can we show in a one flow chart? So we will try to see. Okay. So if you see here, so we can write like this: meiosis. So we have meiosis 1, right? And then we have meiosis 2. So there are events which are differing from meiosis 1 to meiosis 2. And there is a interkinesis state. I mean, interphase is there, which connects both meiosis 1 and meiosis 2. Right? So if you see this meiosis 1, right? Meiosis 1 is something where you can see that there is a recombination of DNA would be taking place. Recombination of DNA taking place, and number of chromosomes reduced to half. If it is 46, it will reduce to 23. Don't always fix to the number of chromosomes 46. 46 number is in the case of diploid human cell content. But in NEET questions, if they give some other cells or some other animal or some other plant chromosome number, don't go with this 46 number. See which species they are talking about, then only you have to answer it. These are the mistakes you can really commit during examination. Okay, so be careful, right? So there is a reduction of the chromosomes, and there is recombination of the chromosomes, right? During M1 phase, but M2 phase, during M2 phase is something which is exactly similar to the mitotic phases. So in this you will be finding, except the prophase, especially the cell directly enters into this meiosis 2, where it quite resembles the mitosis. So mitosis, you have PMAT-C, prophase, metaphase, telophase, anaphase, telophase, and finally cytokinesis. Right, so the two cells which are formed would be enter into this meiosis 2 to form 4 cells. So whatever the genetic component here already fixed would be, this cell will undergo mitosis. That's it. So that's the very easy aspect. So the difference is when you draw a difference here, there is no recombination. Here there is no reduction in chromosome. It means chromosome number would remain the same. Okay, once these cells divide, see, it will be divided into 23, 23, 23. You will find these all cells would possess 23 chromosomes. Okay, so this is equational division. The second aspect you can remember is equational division, equal division. And this M1 phase, you can make it as reductional division. So in this way, if you read, it will be more easy, right? So let's further go into the what do you say? Are you able to follow? Is it clear, guys? Right. Right? Okay, fine. So let's move on to the phases, important phases that you see in meiosis 1, prophase 1. Right? If you are able to see this portion, right? So that is enough. So what you can do is, let's have a small. So this is an important thing, and then we can finish this lesson. Okay. So in mitosis, you need to focus on in which phase this nuclear membrane is dissolved. Right, prophase 1, when the cell is entering into prophase 1, the cell, the nuclear content would get condensed. So I always explain this condensation, right? So condensation is something, condensation of chromosome, we already talked in molecular basis, how this condensation takes place. So this thread-like chromatin material would be eventually condensed to form chromosomes like this. Isn't it? So in M phase, it looks, the chromosomes would look very prominent, right? Very clear. So that would be one of the question. So in previous year's questions, if you see that 32 years previous year's questions I'm posting every day, right? See them, then you will get an idea. Oh, these type of questions they will be asking. Okay, in AIPMT at the same time NEET both I'm giving. So please do take care of that. So this is condensation takes place, and then further it enters into the, what do you say, a division. So here you need to focus on the chromosome structure of the chromosome, and it will have this centromeres will be having, and during this at the centromeres you will be finding, yesterday we talked, right, different based upon the centromere position you will be having different shapes of the chromosome, like J form, V form, L form, V form will be there. So when this V form will be formed, right, when the centromere is, if you see the structure of the chromosome, you can see that this chromosome would contain kinetochore, a plate-like structure. I have shown very beautiful diagrams during the lesson also. Okay, this kinetochores, plate-like structures where the spindle fiber would be attached, and the spindle fibers are microtubules which are formed, the centrosome centrioles, right, which are formed during the G1 G2 phase, right. They will be pulling these chromosomes to one end, poles, right. So during that pulling of these chromosomes, because it is an equatorial division, especially this mitosis is concerned, the chromosomes would move and eventually they form different shapes. So when it is pulled, so when it is pulled, right, so what happens is it forms a structure like this. So it is something which looks like V shape. So this is based upon the centromere. So the kinetochores, their functions, the kinetochore plate, that is red plate, which is attached to the both the sides of the chromosome. Okay, it is not round. Centromere is round actually. It covers the entire chromosome, but the kinetochore looks like a plates, two plates. So either of the sides would be there. So based upon that, the chromosome would be pulled. So this is mitosis. So during prophase, what happens during M phase, that is we are talking about P-mat C. So in this pre-mat C, the prophase events, especially the nuclear membrane, where the nuclear membrane would actually disappear. Again, when it will reform, those things you need to focus. Okay, is that clear? So that is about mitosis. So in mitosis, this anaphase, M phase and anaphase is more important, because during anaphase only the different structures of the chromosome would be formed, because they are pulled towards the poles. So that is about mitosis. It's very simple. And this mitosis is as similar as the meiosis 2, equidirectional division. So once at the end of the meiosis 1, reduction of chromosomes takes place, and then that would be eventually divided further equally divided into forming 4 gametes. But in the case of gametogenesis, oogenesis, three of the ovums which are formed would be polar bodies. They will not have cytoplasm. They will be having only nuclear content, so they will be eliminated. So according to NCERT, they gave only 2 polar bodies, but eventually that one of the polar body also can undergo division, and they can form 3 polar bodies and 1 ovum. That ovum would actually eventually undergoes meeting of the sperm, and then eventually starts its secondary meiosis and to form a definitive ovum. That this is how you need to connect the topics. Okay, so that is about the mitosis and meiosis. And finally, now let's come to the meiosis phases. So what are the phases that you see in meiosis? So if you see meiosis phases, we have leptotin, right? Is that clear, guys? So here in meiosis 1, we have prophase 1, right? Just like PMAT, we have meiosis, we have different phases within the, meiosis. So if you see in NCERT also, they clearly mention what are the different phases that you see in meiosis 1. So in meiosis 1, you will be finding prophase 1, metaphase 1, anaphase 1, telophase 1. And then meiosis 2, again the same thing, PMAT, right? That is, and then followed by cytokinesis, right? So within the meiosis 1 is the where you will find the recombination, right? So where exactly you will find what are the things that are involved here is what you need to focus, right? So if you see here, during this meiosis 1, we have prophase 1 is one of the subphases in this meiosis 1. In this meiosis 1, prophase 1, it is divided into leptotin, zygotin, pachytin, diplotin, and diakinesis. So what are those? Leptotin, zygotin, pachytin, diplotin, and diakinesis. So here kinesis is there, right? Kinesis is something which is splitting, right? You can see that we'll see what exactly happens. So what exactly happens is the leptotin. Leptotin is in prophase 1, right? So there should be condensation should occur to even exchange the genetic material between the homologous chromosome. Here you need to remember the one of the important aspect is I'll use two colors, red for maternal and blue for paternal. Paternal means father side chromosomes, right? It means the chromosomes came from sperm, and maternal, the chromosomes which are present in the ovum. So during fertilization, this maternal and paternal chromosomes forms a diploid zygote and then the diploid organism, and that undergoes meiosis to form haploid gametes. This is a cycle, right? So here I am using what is it? Okay. So let's take this blue as paternal, okay, and let's take this red as with centromeres as maternal. So to signify, to symbolize it, I'll use these symbols. Okay, female, maternal, and paternal. We have male. So this is I'm taking only one set of chromosome, guys. Okay, you need to ascertain this entire thing for the entire 23 sets of chromosomes. So I'm taking one set. Let's take this is chromosome number 1. So if you see this, if you assume this is a chromosome number 1, okay. So we'll see how this chromosome number 1 would recombine. Okay, because now what you see your your genetic composition, it's not it is from your mother as well as father. It is both the traits are combined, right? So likewise, how that combination takes place at the genome level, at the chromosome level, where during this prophase. That's what this prophase is very important. So during the leptotin, what happens is condensation of the chromosomes takes place because it needs to undergo this exchange of genetic material, and then it needs to enter into the mitosis, right? Mitosis 1, right? So that's what the condensation takes place, and the nuclear envelope dissolves, and these are the different events that occurs during this leptotin, right? And further, what happens is once this next it enters into the next phase, that is zygotin. So during the zygotin, synapses would form. So synapses formation would be taking place. So synapses would be forming, or you can see that synaptonemal complex would be developing. So synaptonemal complexes means, let's draw a chromosome like this. Okay. So these are chromatids. So that's what I'm saying. These two are actually what we call as homologous chromosomes. Homologous chromosomes. It means both are the same number of chromosomes. I mean, same type of chromosome. For example, chromosome number 1, chromosome number 1, but one from maternal, one from paternal. That's why we say homologous chromosomes. And within this chromosome, this chromatids are there. Na? These are called sister chromatids. Within the chromosome, for example, if you consider me, we have 2 legs and 2 arms, right? Limbs. So you can consider that the legs and the arms that you possess, imagine that those are chromatids, okay? And these are sister chromatids. These both are sister chromatids, belong to you. And suddenly one person came and you wanted to shake the hands. So when you're shaking hand, this chromatid, and assume that the other chromosome which is having the similar characteristics. If I name myself as chromosome number 1, and there is one more chromosome number 1 comes, okay, my partner, and if we shake hands, what happens is my chromatid, my hand and her hand would be shaking. So it means there is once a shaking hand, right? So the shaking hand is in between the non-sister chromatids. This is very important. So this complex would form between non-sister chromatids. Is it clear? So this is important aspect. I don't know others how they learn, but this is what you need to focus. Is it clear? Sister chromatids and non-sister chromatids. I'm just quickly spending much time here because if you don't understand this, the entire meiosis is gone. That's why I'm spending much time in this. Okay. So once, so let's take this out. Now you understand, right? So this is chromosome number 1 of paternal side, and there is a maternal side chromosome. Your arms and your limbs, the entire limbs, the 4 limbs you have, is sister chromatids. And when you are pairing with someone, those will be paired with, when you shake hands with others, it would be a non-sister chromatid. So let's draw like this. So there should be a genetic material exchange should occur between non-sister chromatids. Okay. Before the exchange itself, there should be synapses formation. It means they should come close and they should form a synaptonemal complex. So they form a complex like this. Okay, they form a complex like this. So here it is there, man. Let's do this. You can see here. So this is synaptonemal complex. Synaptonemal complex. So synapses would begin in the zygote, right? And then it will form bivalents, right? And then it will be entering into the next phases, right? So I have to keep continuing the same color, right? So during the packeting, what happens is crossing over takes place. How that looks like? So you have paternal chromosome and maternal chromosome. Okay, this is paternal chromosome and this is okay. So here what you see is there is a crossing over is taking place. So they are forming X-shaped crossovers, right? So here the exchange would be taking place. So one piece of this material would be going into the maternal chromosome, and one piece of means the DNA sequence would go into the maternal. So there is an exchange of genetic material, a recombination taking place because of formation of crossing over. So during this DNA exchange by non-sister chromatids would be taking place. Is that clear? We are almost came to an end. Once this meiosis 1 is done, you can revise by yourself. Okay, this is an important aspect that even a few students asked me to explain during our NEET preparation also in school. Is that clear? Shall I continue? Is that clear? Are you able to understand this? You already studied about all these things. I'm just revising, that's it. Is that clear? Um, guys, please do respond. We have only 10 minutes time. Yeah, fine. Okay, thank you. Then we'll continue. So here, what happening is during packaging stage, so they are forming crossovers. So what exactly given in your textbook, right? If you see here, bivalent formation. So tetrad formation already taking place, right? During the during the packaging itself. I mean, this during the zygote, bivalents are already formed, and the bivalents once formed, and then during the packaging stage, this exchange of the genetic material would be taking place, forming the crossing overs, right? The crossing over takes place during the packaging, right? And then you will be next you will be finding this chiasmata formation, right? So once this crossing overs leads to recombination of the genetic material, right, of these two chromosomes. Then the recombination between these homologous chromosomes will be completed by the end of the packaging. So once the packaging is done, this exchange of genetic material takes place. I will change the colors also. You just try to see. So at the beginning of the diplotin, what happens is we will be recognizing the dissolution of the synaptonemal complex. So the synaptonemal complex which is formed here, right? So which is formed, the synaptonemal complex would be dissolving, right? So how it would look like, right? Once this is dissolved, how it would look, okay? So it during this, the next phase, right? So So there is an exchange of genetic material already taking place. So you can see that there is a change in the color. Okay, and you have so there is some exchange in the genetic material is already happened, right? And there is a dissolution of this synaptonemal complex. So the synaptonemal complex which is there, it is dissolved, right? Which is dissolved, and what happens is here it will move this this, and it will have a tendency of this recombinant homologous chromosomes to separate, right? So they try to separate from each other at the sites of crossover. So when they are trying to, now first they are like this, when they are trying to separate now at the crossovers, they form this X-shaped chiasma. So they will be forming this X-shaped chiasmata. So this chiasmata formation, X-shaped chiasmata formation will occur in which phase? It is in diploid, it is in diplotin phase. So this X-shaped structure called as chiasmata or chiasmata would be formed. So here is the stage where in oocytes of most vertebrates, the diplotin can actually last for many months or years. So it can actually stop at this phase also. It can initially. And finally, what happens is, that's what now we have seen in oocytes, right? Once the magic touch of this sperm, this plasma membrane of the sperm occurs, it quickly undergoes the meiosis too. I will remember. Then it becomes the mitosis, the secondary oocyte, right? So in that way, already recombination is done. But this excess chiasmata and then chiasmata, the excess chiasmata would be formed, and at the final stage of the meiotic prophase 1, what happens is diakinesis. Kinesis is something breaking. So breaking of the chiasmata. So once this chiasmata is like this, right, and that would be break. So once they break, how they appear? So if you see this during the last phase of diakinesis, they may appear, right, which so it leads to the separation of chromosomes after the exchange. How they may look like. So let me draw the final diagram. Okay. So now The exchange of genetic material already took place, right? So how can we represent? We can represent in this way. Understood? Is it clear? Yeah. So this is how the exchange of genetic material from this red chromosome, that is maternal, is exchanged to the paternal, that is father chromosome. So there is a mixing of the characters would express. So how is that determined? Because this will undergo further reduction division, right, at the end of the meiosis one, right? So still the meiosis one is not yet completed, guys. So this is end of the prophase one, diakinesis. Then it will enter into M1 phase, A1 phase, and T1 phase. Right, during T1 phase, this separation of chromosomes takes place, reduction would take place, and formation of two cells would be formed. So after the two cells are formed, the meiosis 2 would start. So once the meiosis 2 starts, what happens? These chromosomes, right, these chromatids, the 4 chromatids are there right now. Yeah, so these 4 chromatids would act eventually as a chromosomes in the gametes. So if I start drawing the gametes directly, okay, how they be appearing? So if I draw, so it will pass into the next phases, that is M1 phase, okay, P1 phase is done, M1, A1 and T1, and then it enters into M2 phase, okay. After M2 phase, final one is again P-maxi occurs, and at the end of the thing, you will be finding 4 different cells would form, okay. Let me take different color, okay. So 1 cell, 2 cell, 3 cell, and a 4th cell. Okay, so each cell would contain, so here we have chromosomes actually, but it would be reduced to half after the exchange, and then followed by mitosis. Then they would be appearing like this. So here there are normal, parental types as well as a recombinant types. That's what linkage etc. would come. Here we are not talking about linkage and all, but here you may find one of the recombinant chromosome. Okay, and here you can find one of the recombinant chromosome. So if this gamete, right, assume this is sperm. Okay, if this gamete fuses with ovum, this characters, it means mother also has certain characters, those characters went, and father characters also mixed with this ovum. Like this, there is a recombination and the variation is created. That's what I may appear or may be similar to the father, but I'm not exactly the same because the recombination takes place during the meiosis one. So from here you can study, self-study by yourself that what is the significance of meiosis, significance of mitosis. They would be asked as correct or incorrect statements. I hope you enjoyed this class and it will be useful. Okay, please leave feedback in the class group itself. If you have any doubts, right, you can approach me. Is that clear? So you have to focus on the phases and their their characteristics features. At the same time, checkpoints, CDC protein checkpoints, cell cycle checkpoints, mitosis, anaphase and chromosome shapes, centromere, positions, names, metacentric, submetacentric, telocentric, etc. And then kinetochores, their functions, centrioles and their functions. At the same time, meiosis 1 and prophase 1 is the main important aspects. Where does this chiasmata forms and where bivalence forms or divalence forms, and where exactly chiasmata breaks during diakinesis, and then reduction division, and then equatorial division, finally formation of gametes and the significance. That's it. So we'll meet in the next class. Transport in plants is very tough chapter in the entire bio syllabus of the 11th standard, right? very tough. Even 12th standard also, you compare 12th and 11th portions. Transport in plants is very tough chapter, but very easy to understand if you really focus on it. I'll try to explain tomorrow. Water potential concepts, etc. Will be explained tomorrow. So meet you in tomorrow class, and please stay home and stay safe. Take care. I hope you enjoyed this class. Thank you. So tomorrow we'll meet you with the new lesson. Okay. Thank you.